Hello, my dear students. Welcome back to my channel Science with Sangati. So, I hope all of you are doing so great in your life. Today, we are going to discuss further organelles which are remaining, like lysosomes, vacuoles, plastids, mitochondria, cytoskeleton, cilia, and flagella. Okay, so please do watch the complete video if you want to know about these topics. So, let's get started. In the previous video, in the previous lecture, we covered like Golgi apparatus, endoplasmic reticulum, ribosomes. We have covered these, right? And we know that how the proteins were synthesized. Now you can see that Golgi's functions. One of the function is it is helping in the formation of lysosome. Okay, so Golgi apparatus is forming the lysosome also. Now, what is lysosome? Lysosome is an organelle which is also called as suicidal bag. Okay, now why it is called a suicidal bag? You can see in this picture also, this is how it looks like. This is the outer membrane, and inside there is the space which is called as lumen, and in the lumen there is the enzymes are present. Different types of enzymes are there, like digestive enzymes, proteins are also there. Okay, so now these enzymes are called as hydrolytic enzymes. They are acidic in nature. Okay, now these enzymes are helping to break down some macromolecules, okay, like proteins, lipids, carbohydrates, nucleic acids. Okay, these are helping to break it down. So basically, these acidic enzymes, for example, carbohydrates, nucleosidases, okay, lipases. So all of these are the hydrolytic enzymes, and there are almost around 60 types of hydrolytic enzymes are present. Yeah, that many. You don't need to remember so many names. Okay, you can just remember few names like lipase, and carbohydrates. All right. Then protease. So these enzymes are breaking the macromolecules like proteins, lipids, carbohydrates into smaller and smaller particles. And basically, whatever foreign particles entering the cell, foreign particles like bacteria or any pathogen, if they enter the cell, so the pathogens are also made up of these macromolecules only, right? So they just digest those pathogens also. So basically, they break down the pathogens, the surface, the covering of the pathogen, they break it down. So that's how they kill the pathogen. Okay. And not only that, like, uh, not only the pathogen. So suppose if there is any cell organelle, suppose any mitochondria or ribosome, which is not functioning, which is not working, they are present inside the cell. So like these hydrolytic enzymes secreted by lysosomes are also going to digest those cell organelles. So basically, lysosomes is cleaning the cell, you can say. Okay, and when it is cleaning the cell, this is releasing the enzymes, and when it releases the enzyme, sometimes it's burst itself, it is bursting itself and releasing the enzyme, so it is killing itself also. Okay, so that's why it is also called as suicidal bag. It gets burst and it releases the enzyme, and those enzymes further digest the other unwanted particles which is present inside the cell. Okay, they also participate in the programmed cell death, which is also called as the apoptosis, by degrading the cellular components. Okay, so these are the functions of lysosome. Now, next cell organelle is the vacuole. Vacuole is mainly present in the plant cell. Okay, why I am saying mainly? Because in the plant cell, one vacuole is present, which is very large and very prominent. In the animal cell, vacuole may be present or may not be present. It is that not that much required. Okay, and if it is present, they are very smaller in size and they are multiple in number. Not one, there will be multiple small size vacuoles will be there in the animal cells. And in some animal cells, vacuoles are not even present. Okay, so now what is the function of this vacuole? Vacuole is kind of storage. Okay, so you can say that vacuoles are the storage bag. All right, or storage sacks, you can also say that. Okay, now this vacuole is storing the all materials. Like what you do if you have any leftover food, where you are going to store it? You are going to store it on your refrigerator, right? Inside the refrigerator we store the leftover food. But where plants are going to store it? So plants will be storing it in the vacuoles. Okay, so like it is storing the water here, water over here. There could be any organic, organic minerals are present in the vacuole, nutrients are present, ions are present, okay? Any waste products are also present inside the vacuole. And all of these together are called as cell sap. Okay, so cell sap is not only the water, but water plus all of these mixtures, proteins, minerals, organic acids, all of these together are called as cell saps. Okay, now as this cell sap is a fluid substance, so this is going to create a pressure on the outer part of the vacuole, outer membrane, and the outer membrane of the vacuole is called as tonoplast. Okay, the membrane of a vacuole is called as tonoplast. So this is going to create a pressure on the outer wall. It's very similar, like you take a balloon and you inside the balloon you fill it up with the water. Okay, like we play a holy balloon, right? So we play insert water inside the holy balloon. So what will happen? The water is going to create a pressure on the surface of the balloon, right? So the balloon is now very tough. It's a kind of turgid. So that is called as turgidity. So balloon takes a proper shape. So that is called as turgidity. So inside the cell sap, when all of these like inside the vacuole, when the cell sap is present, this is taking a proper shape. It is becoming tight. Okay, now, as I told you that in the plant cell there is one vacuole and that also very large, so it will look like this. Okay, so you can see that maximum volume of the plant is covered by the vacuole only, and that is 90% of the cell volume is covered by the vacuole, almost 80 to 90%. So that means when the cell sap is applying this pressure, which is the Turger pressure, of course this pressure will be applied on the wall of the cell also, right? The plasma membrane of the cell. So it is going to maintain the shape of the cell. Okay, so this turgor pressure is very helpful for the plant in maintaining the shape and also maintaining the osmotic balance of the cell. Okay, you can easily see that when you are watering the plants, the leaves are very fresh, right? They are flourished. They looks very tightened up in their original position, right? But if you do not water your plants, suppose for a week, what will happen? The leaves will get wilted, right? So that is because there is a lack of turgor pressure. The leaf gets wilted, and that is called as flaccidity. Clear? So this is the vacuole. Vacuole also do contain some hydrolytic enzymes, okay, which is helping in the digestion of some unwanted macromolecules. So these are the functions of the vacuole. Now let's move on to the next cell organ, very important one, which is the mitochondria. The other name of mitochondria is powerhouse of the cell. So this is supplying the power. Okay, this is the powerhouse of the cell. This is supplying power in the form of ATP, adenosine triphosphate. It is a double membrane bound structure. Here you can see in the diagram, this is the mitochondria and this is the outer membrane. Okay, very smooth and it's permeable also. Okay, and the inner membrane is having these foldings which are called as the cristae. Okay, now these cristae are helping to increase the surface area for maximum production of ATP. Okay, now here you can see this the space between the outer membrane and inner membrane is called as the like intermembrane space, and here this space is called as the matrix. Here the jelly like fluid matrix is present, so we call it as a mitochondrial matrix. Okay, so now in this matrix, mitochondrial DNA, which is also called as mtDNA, is present. RNA is also present. Ribosome is also present. Okay, so mitochondria is the organelle which has its own DNA and ribosomes in it. All right, so it can synthesize its own proteins. I hope this is clear to all of you. Now it is very colorless. It do not have any pigment. Okay, so and it is producing proteins. Sorry, it is producing energy, which is in the form of ATP, the energy currency of the cell. Now, it produces ATP especially during the aerobic respiration. Okay, so you will learn that in another chapter about the Krebs cycles, glycolysis and the Krebs cycle, where the ATP is getting produced. Okay, so ATP production, like through a series of biochemical reaction occurring in the inner mitochondrial membrane. Okay, and the mitochondria is producing ATP. So there are like series of biochemical reactions like the glycolysis and the Krebs cycles. Okay, where the ATP is getting produced, and after the Krebs cycle there is the ETS. Apoptosis, mitochondria play a very crucial role in programmed cell death also by releasing some specific proteins and the enzymes. Okay, they also help in the detoxification of ammonia. Okay, uh, so it is a toxic byproduct of metabolism and it is converting it into a less harmful substances. So these are the main functions of mitochondria. So I'm just now going to show you a video. Please do watch this video, okay, for better clarification.
Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes.
All right, so let's discuss about the cytoskeleton. Cytoskeletons are made up of a network of proteins only. So what kind of proteins are there? You can see microtubules, okay, intermediate filaments, and the microfilaments are there. Okay, microtubules are made up of the tubulin protein, microfilaments are made up of the actin protein. So all of these proteins are present in a tube-like structure, same you can see in the picture. They are basically a tube-like structures. All of the structures are tube-like only. Okay. Now, what is the meaning of the skeleton? What is the purpose of a skeleton? So, skeleton is providing a structure to our body, right? So here, the cytoskeleton is also help to maintain the shape of the cell. So see, what are the things which are maintaining the shape? First, we just discussed that the cell wall in the plant is maintaining the shape of the cell, right? Then the endoplasmic reticulum, that is also providing a mechanical support to the cell because the structure of the endoplasmic reticulum is also a very tubular structure, right? Which is connecting the nuclear membrane to the plasma membrane. Then we saw that the vacuoles in the plant, it is also helpful in maintaining the shape by the turgor pressure. Now, the cytoskeleton is also helpful to maintain the shape of the cell. So it is providing a very basic structural support or mechanical support to the shape of the cell. Okay, so it is also facilitating the cell movement and shape maintenance and involved in the intracellular transport and the cell division process also. Okay, so intracellular transport, it is helping because it is a tubular structure, right? So it is helping to transport the, so like the tubular structures like a road. Okay, so through the road what happens? The cars or the vehicles are moving, right? So here also through any tubular structure like the in the endoplasmic reticulum also we have seen it is also helping in transportation. Here also cytoskeleton is also helping in the transportation. Just imagine that it's like a road. Okay, so cytoskeleton main function is these. All right, now let's move on to the next thing, that is the cilia and the flagella. So cilia and flagella are the hair-like extensions. Okay, so flagella are pretty longer. Okay, and if the flagella is present, so it will be lesser or fewer in number, maybe one or two. Okay, but cilia are shorter structure, but multiple cilia are present. So many cilia will be present on the surface. Okay, so that's the cilia. Now here you can see that it is both of these are producing 9 plus 2 arrangement of the microtubules which consist of central pair. Okay, so here you can see in the surrounding there are like 9 and here 2 microtubules. Okay, so this is the basic structure of the cilia or flagella, whatever. Here you can see the dynein arms are there. So these dynein arms, these small little dynein arms here you can see, these are having a very important role. Okay, so they are moving in like a way that it helps to move the cilia or the flagella. So that's how they are helping in the movement of the organisms also. Okay, so flagella here you can see this is having another structure which is called as the body. Okay, so by the basal body is having some hook like structure which is helping to attach it, all right, to any surface. So these are present in the prokaryotes, basically the flagella, you can see the basal body. This is present in the prokaryotic structure and this is the filament structure. Okay, so these dyneins are helping. They are like they are like a hook or leg-like structure, like they are moving like this, and that's how they are helping to in the locomotion of any microscopic organism. I'm going to show you a video which will make you a very clear idea about how they are moving. Let's watch this video.
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized the molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat. As one side is inhibited, the other side is active. In our model, regulatory signals result in inhibited dyneins on one side. This allows dyneins on the opposite side of the flagellum to walk along the microtubules. Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic beating motion. The dynein inhibition may occur in an all-or-none fashion, similar to an action potential, which could explain how cilia and flagella can switch their bending direction back and forth in less than 1/100th of a second and continue beating for days.
All right, now I hope it is clear that how the cilia and flagella are helping to move the organism. Now, moving on to the next organelle, that is the centrosome. Okay, so centrosome is the organelle which are mainly present in the animal cell, and they are helping a crucial role in the cell division process. So here you can see this is the structure of the centrosome. Inside the centrosome, the centrioles are there, which are the cylindrical structures, and the centrosomes are covered by some microtubules. Okay, so these centrioles are again, they are made up of microtubules only. Here you can see the microtubules are there. So they are in the arrangement of three together. Okay, so triplets of microtubules are there. Okay, like a cartwheel patterns are there. So these centrioles are helping in the process of cell division process. They release the spindle fibers, with the spindle fibers then they get attached to the kinetochores, and they help in the separation of those chromatids. Okay, so these are the functions of centrioles and centrosomes. Now let's move on to the other cell organelle, that is plastids. Plastids are present in the plant cell. Okay. Now they are also the organelle which are having their own DNA and ribosomes. Now there are different types of plastids present, like chromoplast. Chromoplast contains the pigments which are called as carotene and xanthophylls. So they provide yellow, orange or red color to the plant cell. Okay, so you can just remember carotene, So carrot is having orange, red, yellow color. So the pigments are orange or red, yellow color. Okay. So they are mainly providing color to the fruits, flowers, and whatever the other structures of the plant which are in orange, yellow, red color. Okay. So these are the chromoplast. Now, next type of plastid is called as the leucoplast. They are colorless plastids and they are also helpful in the storage functions. Okay. So now various types of leucoplast are there, like amyloplast helps to store the carbohydrates or the starch. Okay, elioplast helps to store the oils or the fats. Okay, eleuroplast helps to store the proteins. So these are the different types of leucoplast. Now, next very important plastid is the chloroplast. So chloroplast is having the very important green color pigment which is called as the chlorophyll. Okay, and these chlorophylls helps to capture the light energy and they helps in the light dependent reaction. Okay, so this chlorophyll is present where? So chlorophyll is basically present in the thylakoids. Okay, where so here you can see the structure of the chloroplast. These disc shaped structures are called as thylakoids, and stacks of disc-shaped structure like stacks of thylakoid altogether is called as the granum. So here you can see stacks of thylakoids are called as the granum. Okay. Now these granums are again joined together by a tubular structure. You can see over here these are called as the lamellae. Okay. And inside the thylakoid there is a space which is called as lumen. Okay, inside the chloroplast, there is a fluid present which is called as the stroma, and chloroplast is also a double membrane structure. Okay, so this is the basic structure of the chloroplast. Now, the chlorophyll is present on the outer membrane of the thylakoid. So if I say this is the thylakoid, okay, not the outer membrane, uh, basically the membranous part. Okay, so this is the membrane. I'm drawing it very thick. So here the chlorophyll will be present, not in the lumen. Okay. In the lumen, there is no chlorophyll. In the membrane, chlorophyll is present. So basically the light dependent reaction is happening in the membrane part of the thylakoid only. Okay. So this is the function of the chloroplast. It is helping in the photosynthesis process. Alright students, today we have discussed about the lysosome, vacuole, mitochondria, then centrioles, centrosomes, cilia, cytoskeleton, and the plastids. Okay, so that's all for today's video. If you do like this, please hit the like button and share this with your friends also. Okay, so we will meet in the next video very soon. Okay, till then, keep watching. So take care, have a nice day, all of you. Bye bye.